Hi, and welcome to the urinary system part 2 of 2. In the second half of the urinary system, I'll focus on the ways in which the kidneys are able to modify their behavior in order to respond to moment changes in our blood composition. In part 1, we talked about the general ways in which the kidneys contribute to homeostasis, and we spent a lot of time talking about how urine is formed. And one of the most important contributions of the kidneys is to maintain the composition of blood, to maintain, among other things, the relative concentrations of water versus solutes. You can think of that as osmolarity. The kidneys also are responsible for maintaining blood volume, which is directly related to blood pressure. And so in part 2, we'll talk about how the kidneys are able to adjust their processes, filtration, reabsorption, and secretion, in order to address the body's changing needs. And we'll focus on water balance, so how much water is in your body, what's the relative concentration of water versus solutes in your blood. We'll talk about a pretty complicated interplay between the endocrine system and the cardiovascular system in order to address changes in hydration, how much water is in the body, and how to maintain blood pressure in the face of low blood volume. We'll talk about a class of drugs called diuretics. They all act on the kidney. And micturition refers to the act of urination, and then I'll give you a little bit of information about dialysis and a developing technology, artificial kidneys. Homeostasis is an important theme this semester. It permeates every organ system that we cover. And a very basic question for every organ system is, how does this particular organ system contribute to homeostasis? And there are many different ways to explain homeostasis, to categorize it. I like this particular phrase, that homeostasis is a constant shifting balance, and your kidneys are constantly shifting in all sorts of ways in order to address moment-to-moment changes in your blood composition. And here's this cute little boy on the balance ball, and in order for him to stay balanced, he needs to respond to moment, very slight changes. He's not standing still, but he's making these very micro changes, and that allows him to keep his balance. Your kidneys are always making these little micro changes in order to maintain the composition of your blood. One of the most important functions of the kidneys is to help maintain the proper ratio of solutes to water in your blood, to maintain blood osmolarity and to maintain blood volume. So we're going to talk about the mechanisms by which our kidneys can hold on to body water when body water is scarce, and our kidneys can also eliminate body water when body water is in surplus. So in order for us to exhibit what's known as water balance, the daily volume of water that we take into our bodies needs to equal the daily volume of water that leaves our bodies. So in this table from your textbook, we can look at sources of body water gain, that's our intake, and sources of body water loss, that's our output. So let's start up top with the intake. We ingest, meaning we take by mouth, roughly 1,400 milliliters in liquid and 1,100 milliliters in food. And we also make a little bit of water all of the time during aerobic cellular respiration. We form new molecules of water inside our mitochondria. We have not covered that yet. We would cover that in the metabolism chapter. Regardless, it is a significant source of body water. So water comes into our body through our mouths. We ingest water and we make some new water molecules as part of ATP production. And so in these example figures, our total body water gain per day is 2,850 milliliters. And then that should match our output. And on a typical day, You lose water, roughly 900 milliliters, in the air that you exhale. That air is humid. If you breathe against a mirror, for example, you'll be able to see the condensation of water. You also lose water across your skin through sweating, but insensible water loss is water loss that we don't detect. So you are losing water from your body in your breath and across your skin. And across your skin, even if you're not noticeably sweaty, you're still losing some water to the atmosphere. And then, of course, during physical activity or when ambient temperature goes up, we sweat. That's different from insensible loss. We notice it in feces and in urine. And all of these ways in which the body loses water should roughly total the total water intake. So in this diagram here, we have 2850 milliliters of intake and we have 2850 milliliters of loss. Your kidneys exert a lot of control over how much water leaves your body in urine and how much water is retained in your body, is kept in your body. And in this way, your kidneys influence these three factors: the osmolarity of your blood, which, remember, osmolarity can be viewed as the overall total concentration of your blood when you account for all of the different dissolved solutes. And blood osmolarity is approximately 300 milliosmoles per liter. Your kidneys also exert a huge influence over your total blood volume. What's the total volume of blood that's in your body? And then blood volume is very related to blood pressure. So let's review that relationship or think about that relationship. Remember, your cardiovascular system is this closed set of tubes through which a particular volume of fluid, blood, flows. So in this closed set of tubes, what would happen to blood pressure if we were to increase the blood volume? So as it says on the slide, as blood volume increases, blood pressure blank. Blood pressure is going to increase. If we have a higher volume of fluid circulating throughout this closed set of tubes, then the pressure inside that closed set of tubes is going to go up. And then looking at that second statement there, as blood volume decreases, blood pressure also decreases. If we have a lower volume, less fluid flowing through this closed set of tubes, the pressure inside that system is going to go down. So there are many overlapping homeostatic mechanisms for maintaining blood volume and blood pressure. And we always talk about the two together because they are so interrelated. Blood volume and blood pressure. If something causes blood volume to go up, it will cause blood pressure to go up, and the opposite is also true. And bear in mind that there are mechanisms for independently regulating blood volume versus blood pressure. They can be controlled independently, but they are very interrelated and interconnected. So we are going to talk about this combination of nervous and endocrine responses that involve the nervous system, the endocrine system, the cardiovascular system, and the urinary system. And these are all mechanisms for regulating how much water is in your blood, what's your total blood volume, and what's your overall blood pressure from moment to moment. All right, we'll start with, perhaps it's the simplest of these three mechanisms that we're going to talk about. ANP stands for atrial natriuretic peptide, where the natriuretic means NAT is sodium, right? Like the chemical symbol for sodium is Na. Natriuretic. There's more sodium released in your urine. So ANP is a hormone that's secreted by your heart. Yes, your heart is an endocrine structure. It produces one hormone, and it produces this hormone in response to high blood volume. So here's a question on the slide. Under what conditions is it favorable to get rid of excess water? And one way to answer that question is when there's too much water in the body. So let's say over the next 10 minutes, you ingest 3 liters of water. I don't recommend that, but it would certainly put you into a state of hyperhydration. Those water molecules would land in your stomach and they would cross the mucosa, they would cross the lining of your GI tract, your stomach and your small intestine, and those water molecules would get into your blood and they would have the effect of diluting your blood. So after you ingest those 3 liters of water over a relatively short period of time, your blood volume would go up. What would happen to your blood osmolarity? Your blood osmolarity would go down. More water molecules, fewer solutes. And now we're out of homeostasis. And so it's up to your kidneys to help your body get rid of excess water. And the primary means in which your body does that is mediated through this hormone called ANP. So again, ANP is produced by your heart, and the signal, the stimulus that causes your heart to secrete ANP is the degree of stretch that occurs in the atria of your heart. So inside the atria of your heart, you have these stretch sensitive cells. So when venous return goes up, these stretch sensitive cells detect that there's a higher than normal volume of blood returning to the heart. And these stretch sensitive cells detect that and lead to the secretion of ANP. ANP is a circulating hormone, and that means it gets secreted and it travels all over your body in the blood. So ANP has a couple of different actions, and the action of any hormone is the behavioral change that it brings about in a target tissue. So I'm going to talk about 2 different target tissues here, and both of these target tissues, by definition of a target tissue, they have receptors for ANP and they therefore change their behavior in response to ANP. And the behavioral responses are called actions. And so we'll talk about 2 actions of ANP. One of those actions influences the rate at which glomerular filtration forms. The other response involves the rate at which sodium is reabsorbed in the nephron. One way to think about ANP is this hormone, this signaling molecule, is all about how do we get more water out of the body faster in urine? And one way that we can eliminate more water from the body is to make urine more quickly. And the way we speed up all of the processes that happen in the nephron, well, not all of them, but the way that we speed up the rate at which that conveyor belt moves, the rate at which filtrate flows through the nephron, through the tubules, the way we modify that is by controlling the rate of filtration. So you remember filtration occurs when water and solutes leave the blood and enter the glomerular capillary. When we speed that up, when we form filtrate more quickly, it speeds up the rate at which that filtrate travels through the tubules. So it speeds up the formation of urine. And that necessarily is going to lead to a greater loss of water from the body. If we make urine faster, we're going to lose more water. And so shown here is an illustration indicating how the diameters of the afferents and efferents arterioles leading into and away from the glomeruli, how the diameter of those 2 arterioles influences the rate of glomerular filtration. So let's just start in panel A, so the upper left panel. In the upper left panel, we see that something has caused constriction, a vasoconstriction, a narrowing of the afferents arteriole. How will that influence blood pressure in those glomerular capillaries? So now we've increased vascular resistance in the inlet, the incoming arteriole into the glomerulus. And that's going to limit blood flow through the glomerulus. And so as you can see, there's a little down arrow next to P sub GC. So the pressure, the hydrostatic pressure, the blood pressure inside the glomerular capillary is going to go down. And that's going to have a slowing effect on the rate of glomerular filtration. GFR means glomerular filtration rate. And then we can look over at panel B, so top right, where now we have a normal diameter for the afferent arteriole, but we have vasoconstriction happening in the efferent arteriole. When we limit blood flow out of the capillary, we increase the hydrostatic pressure, the blood pressure inside that capillary. So that's indicated there by the little up arrow next to PGC. That indicates that the hydrostatic pressure of blood against the blood vessel wall in the glomerulus has increased. And when we jack up that blood pressure, we increase the rate at which filtrate forms. So water and solutes are going to cross that filtration membrane faster. That's going to increase glomerular filtration rate, and it increases the rate at which we form urine. And then down below in panel C, which is lower left, we have a normal diameter afferent arteriole, but we have a dilated efferent arteriole. So it's extra easy for blood to leave the glomerulus. And so the hydrostatic pressure inside that glomerular capillary is going to fall. And under lower hydrostatic pressure, that means that filtrate forms more slowly. So dilating that efferent arteriole causes glomerular filtration rate to slow. And then on our fourth panel here, panel D, lower right, we see we have a dilated afferent arteriole that lowers vascular resistance. It makes it easier for blood to flow into that glomerular capillary. And we see that the pressure, the hydrostatic pressure, the blood pressure inside that glomerular capillary goes up. And as indicated by that big fat arrow, we see that that also raises glomerular filtration rate. And so the role that AMP here plays here is that it's able to induce dilation of the afferent arteriole coupled with constriction of the efferent arteriole. So if we make the inlet wider and the outlet narrower, that's going to dramatically increase the blood pressure, the hydrostatic pressure, P sub GC. It will be dramatically increased, and that will lead to a faster rate of glomerular filtration. A second mechanism by which ANP causes the rate of glomerular filtration to increase is via relaxation of the mesangial cells. We've not yet talked about mesangial cells. Mesangial cells are shown on this diagram. They're in yellow, and they are modified smooth muscle cells. And remember, smooth muscle cells, or muscle cells in general, what muscle cells do is contract and relax. And when they contract, they pull on their points of attachment. We'll look at where these muscle cells are situated. These muscle cells are attached to different points along the length of these glomerular capillaries. And so when these muscle cells relax, these mesangial cells, when they relax, they allow the capillary loops to spread out, to fan out. And this means that the capillary loops are not necessarily touching each other, but they're spread apart. And this allows for greater surface area for filtration to occur. So when mesangial cells relax under the influence of ANP, that also augments the rate of glomerular filtration. Another method by which ANP helps to get rid of excess body water is by reducing the reabsorption of sodium in the proximal convoluted tubule. So you remember osmosis. Where the solutes go, water flows. Water always follows solutes. So now our objective here is to keep the water in the renal tubule. We want much of that water to leave the body as urine. And in order to do that, we need to prevent a lot of that water from being reabsorbed. And one way to do that is by limiting the reabsorption of a particular solute. In this case, it's sodium. So an action of ANP on the tubule cells that line the PCT is to slow the rate at which those tubule cells remove sodium from the filtrate and return it to the blood. That's reabsorption. So if we slow the reabsorption of sodium, we're necessarily going to slow the reabsorption of water. So more sodium will stay inside the tubule, more water will stay inside the tubule. And this is going to lead to a larger volume of urine, more urine output, and that will help lower blood volume, which will also help lower blood pressure because remember, those two things are related. So in this homeostatic mechanism shown here, we can start with the change in homeostasis, right? Our initial problem is that our blood volume is too high, right? Because what was our scenario? You drank 3 liters of water in a span of 10 minutes, and that water entered your blood, and now your blood volume is too high, and our bodies need to shed excess water. Well, what's that homeostatic mechanism? When we have high blood volume, that's going to lead to an increase in venous return, the volume of blood returning to each of the atria. And there are stretch sensitive cells inside the atria of the heart, and they detect this higher than normal venous return. It's an indicator that blood volume is too high. And cells in the heart respond to this increase in stretch by secreting ANP. ANP has the overall function of increasing sodium and water excretion, right? So we eliminate more water, we eliminate more sodium, and that has the effect of increasing urine volume, which will then decrease blood volume. And notice that this is a negative feedback system. As blood volume returns to normal, those stretch-sensitive cells are no longer going to be stretched to the same degree, and so they will stop secreting ANP. In this diagram, we can review the ways in which ANP acts on the kidney to help eliminate more water from the body. So to review, ANP increases the rate of glomerular filtration. It speeds up the rate at which filtrate forms and therefore speeds up the rate at which urine is produced. 2 ways. Number 1, ANP can alter the diameter of the afferent and the efferent arterioles in such a way that it dramatically increases the hydrostatic pressure inside the glomerular capillaries. And when there's high blood pressure inside those glomerular capillaries, that's going to dramatically increase the rate at which filtrate forms. A second method by which ANP increases glomerular filtration rate is by causing those mesangial cells to relax. The loops of the capillary spread apart and glomerular filtration rate is increased. And then a separate mechanism is shown here as well. ANP decreases the rate at which sodium is removed from the filtrate and returned to the blood. So as we leave, essentially we leave more sodium in the filtrate, and that means we necessarily leave more water in the filtrate. And so that extra sodium and extra water is going to end up in the urine and be excreted from the body. We're interested in how the kidneys are able to change their behavior in order to either get rid of more water or hold on to more body water. So we just finished talking about hormone number 1 here, ANP, atrial natriuretic peptide. ANP helps the body eliminate excess water. Mechanisms, or hormones, I should say, hormones 2 and 3, or mechanisms 2 and 3, shown here, they have similar goals to each other, and the goals are opposite to that of ANP. The RAA pathway and vasopressin, which is also known as ADH. These mechanisms aim to hold on to body water, to not excrete more than we have to as urine, in an attempt to maintain blood volume and raise blood pressure. So we'll start with the RAA pathway, where RAA stands for renin-angiotensin-aldosterone pathway. It's a fairly complex pathway, that affects the cardiovascular system and the urinary system and the endocrine system. So the RAA pathway gets triggered, it gets engaged as a response to low blood volume and low blood pressure. So there's not enough water in the body. Either the person is moderately dehydrated or perhaps there's been blood loss. And so our goal here is to hold on to body water and also to raise blood pressure. The way in which the RAA pathway attempts to hold on to body water and raise blood pressure are shown here. First, the RAA pathway is going to lead to the production of a particular signaling molecule called angiotensin II. And angiotensin II exerts an influence over many different tissues in the body. We're going to talk about how angiotensin II addresses these 3 bullet points here. Angiotensin II is going to help reduce the rate of glomerular filtration. So when we slow down GFR, we slow down the rate at which urine is produced, and we necessarily therefore slow down the rate at which water is lost from the body. Second bullet point, angiotensin II, which is part of that RAA pathway, is going to increase obligatory water reabsorption. So it's going to cause the kidneys to reabsorb more water out of the filtrate and move that water back into the blood to hold on to more body water. And then the third bullet point doesn't affect the kidneys directly, but rather that signaling molecule called angiotensin II is going to cause vasoconstriction of systemic arterioles. So all over the body, many blood vessels would become narrower. And remember, we have this relatively fixed volume of fluid flowing around this closed system. So if the pipes get narrower, that will increase the pressure in the system. So the RAA pathway has many players, and ultimately we want to get to the formation of this signaling molecule called angiotensin II. Well, how does that happen? The RAA pathway is triggered by particular cells in the kidney, the juxtaglomerular cells. When the juxtaglomerular cells detect that there is insufficient blood flow, and this is caused by low blood volume, low blood pressure. When these juxtaglomerular cells detect low blood flow, they respond by secreting an enzyme. It's not a signaling molecule. It's an enzyme called renin. They secrete renin into the blood. And renin acts on a substrate. It's an enzyme. All enzymes act upon a substrate. The substrate for renin is a precursor molecule called angiotensinogen. There's that suffix again, ogen, that we talked about recently. So whenever you see the suffix, when you see a word end in ogen, it's a good guess that that molecule is some type of precursor. That molecule itself doesn't have an effect, but with a very minor chemical change, it will become an activated molecule, whether it's an enzyme or a signaling molecule. So angiotensinogen is a plasma protein. It's in our blood all the time. It's produced by our liver. Angiotensinogen is inactive. It doesn't do anything. It doesn't cleave anything. It doesn't bind to any receptors. But when angiotensinogen encounters the enzyme renin, renin acts on angiotensinogen and converts it into a different molecule called angiotensin I. Angiotensin I is also an inactive precursor. And angiotensin I will be converted into the very active signaling molecule called angiotensin II by an enzyme known as ACE. So ACE stands for angiotensin converting enzyme. And it's a membrane-bound enzyme. So it's a protein that's expressed by a variety of cells, tissue types in our body, and this enzyme exists as part of the plasma membrane. And one of the most abundant locations of ACE in our body is in our lung tissue. It's not the only location, but it is a particularly rich source of ACE. So as blood that contains angiotensin 1, as that angiotensin 1 passes through pulmonary capillaries, it is converted into angiotensin 2 by angiotensin converting enzyme, which is a protein that's part of the plasma membrane of endothelial cells. So the cells that are lining those pulmonary capillaries, they express this enzyme, and as that blood rushes past the cells, the enzyme is able to act on its substrate. The substrate for angiotensin converting enzyme is angiotensin 1, and the product is angiotensin 2. Angiotensin 2 is a signaling molecule that affects many different tissues in our body. And the overarching goals here with the formation of angiotensin 2 are to maintain blood volume, meaning don't lose more body water than you have to, and raise blood pressure. So angiotensin 2 is a hormone. It's a circulating hormone. It's in our blood and it acts upon many different tissues. I'm going to present you with 4 actions of angiotensin 2, and even that is not a comprehensive list. This is a simplified version of the effects that angiotensin bring about. So our first effect of angiotensin 2 is that it slows the rate at which glomerular filtration occurs. And the way angiotensin 2 accomplishes this is by modifying the diameter of the afferent and the efferent arteriole, leading to and away from the glomerulus. So if both of these blood vessels vasoconstrict, we'll limit blood flow through the glomerular capillaries. By constricting the afferent arteriole, we limit blood flow into the capillary, right? And that will necessarily cause the hydrostatic pressure inside the glomerular capillaries to drop. But we need to maintain that hydrostatic pressure at some level because we always have to be producing urine. Even if we're producing urine slowly, our cells are constantly adding waste products to the blood. So urine necessarily needs to be formed all the time in order to get rid of those wastes. But we can slow the production of urine by slowing the rate of glomerular filtration, by vasoconstricting the afferent and the efferent arteriole. The second mechanism by which angiotensin 2 helps the kidney, helps the body hold on to body water, is by causing greater reabsorption of sodium and chloride, and water will follow, right, in the proximal convoluted tubule. If we pull more solutes out of the filtrate and return those solutes to the blood, water has to follow by osmosis. Water follows solutes. So if you want the water to go somewhere, move the solutes and the water will follow. So in the proximal convoluted tubule, under the influence of angiotensin 2, those cells are more actively taking up sodium and chloride out of the filtrate, putting those solutes into blood, and water will necessarily follow. So this is increasing obligatory water reabsorption. So now we have less water, fewer solutes, less water in the filtrate. So this is another means by which we slow down the rate of urine formation and we hold on to body water. Another mechanism by which angiotensin 2 acts to hold on to body water and raise blood pressure is to promote vasoconstriction of systemic blood vessels. Both arterioles and veins can vasoconstrict or venoconstrict. And again, when we narrow the diameter of those tubes, the pressure inside the system will go up. And so here's that schematic diagram of both pulmonary and systemic circulation. So for those systemic capillary beds above the heart and below the heart, imagine that those vessels get narrower. And so we have this closed system and the pressure is necessarily going to go up. So angiotensin 2 is a powerful thing that raises blood pressure. And then finally, the fourth action of angiotensin II that we'll cover is that it stimulates the release of another hormone called aldosterone from the adrenal glands. We've talked about the adrenal glands before, only we've talked about the adrenal medulla, and today we're going to talk about the adrenal cortex. So your adrenal glands are really two glands in one. At the core of the adrenal gland is the medulla, and the medulla is made up of those chromaffin cells, those modified postganglionic sympathetic motor neurons that are really neurosecretory cells. In response to an increase in sympathetic stimulation, the cells that make up the adrenal medulla release epinephrine and norepinephrine into the blood. That's what we've talked about previously. Today, I'm going to introduce you to the adrenal cortex, totally different gland, even though it's packaged together with the adrenal medulla. The adrenal cortex is made up of a different selection of cells, and they're not directly tied to the sympathetic branch of the autonomic nervous system. And the adrenal cortex releases a variety of signaling molecules that one way or another play a role in how the body uses or conserves molecules that it already has. So the adrenal cortex in general plays a role over various aspects of metabolism, how your body uses particular molecules. And here we're interested in a particular cortical hormone called aldosterone. Aldosterone is a hormone, a circulating hormone, a signaling molecule that is secreted by cells in the adrenal cortex. And aldosterone has several different effects, several different actions. The two actions of aldosterone that we'll cover is that aldosterone also contributes to enhancing obligatory water reabsorption in the kidney. It causes the kidney to reabsorb more sodium, more chloride, and more water. So it has a similar action on the kidney as angiotensin II, but aldosterone acts on a different set of cells in the renal tubules. And then the second action of aldosterone that we'll talk about is that it promotes the secretion of potassium into urine. So aldosterone has this secondary role. It plays a role in blood potassium homeostasis, which is sort of indirectly related to what we're focusing on now. Right now, we're focusing on the RAA pathway and the role that angiotensin II plays in holding onto body water and maintaining blood pressure. One of those ways is by stimulating the secretion of aldosterone from the adrenal cortex. And aldosterone acts on the kidneys to increase obligatory water reabsorption. I love this diagram. The RAA pathway is a complicated thing, and this diagram allows us to review where we've been. And so let's just go through this diagram from left to right, starting with angiotensinogen on the left side of the diagram. So angiotensinogen is a plasma protein. It's in your blood all the time. It's produced by your liver, and it doesn't have any activity. It doesn't cleave anything. It doesn't bind to anything. It is an inactive molecule. And that angiotensinogen is only activated when it encounters an enzyme called renin. Renin is an enzyme that's secreted by particular cells in your kidney when those kidney cells detect low blood flow and low blood volume. So the kidneys aren't getting enough blood, and these kidney cells release renin. Renin is an enzyme, and its substrate is angiotensinogen. So in your blood, when angiotensinogen encounters renin, it gets converted into angiotensin I. Angiotensin I is also an inactive molecule. It doesn't do anything. It doesn't cleave anything. It doesn't bind to any receptor. But angiotensin I will be converted into angiotensin II whenever it encounters an enzyme called ACE, where ACE stands for angiotensin converting enzyme. It's a good name, right? So ACE is a membrane-associated protein. So it's inserted into the plasma membrane of cells located in a variety of different places in our body. But one of the most abundant sources of ACE is in endothelial cells in the blood vessels of our lungs. So as blood that contains angiotensin 1, as that blood passes through our pulmonary vessels, it passes through blood vessels in our lungs, it's very likely to encounter ACE. And ACE converts angiotensin 1 into angiotensin 2. And I hope you can see that there's sort of this starburst spray of blue dotted lines radiating out from that word on this diagram. And that tells us that angiotensin 2 has many actions. Angiotensin 2 is a signaling molecule that influences many different tissues in our body. And overall, angiotensin 2 works to hold on to body water and to raise blood pressure. So there are more arrows on this diagram than we have or than we will talk about. So let me just review the things that we actually have talked about. The effects of angiotensin 2. So we have talked about how angiotensin 2 increases the tubular reabsorption of sodium and chloride, and that necessarily increases the tubular reabsorption of water in the kidneys. So angiotensin 2 helps limit the volume of urine by causing an increase in obligatory water reabsorption. Where the solutes go, water flows. Angiotensin 2 also acts on the adrenal cortex to stimulate the secretion of a separate signaling molecule known as aldosterone. Aldosterone also affects the kidney, so maybe you can see this arrow going up. Aldosterone further promotes the reabsorption of sodium, the reabsorption of chloride, as well as the as well as the excretion of potassium. But really, we're most interested here in the reabsorption of ions, which promotes the reabsorption of water. So we're holding on to more body water. Another action of angiotensin 2 that we've talked about is right here. Angiotensin 2 is a vasoconstrictor. So it triggers vasoconstriction in blood vessels all around the body, and that powerfully raises blood pressure. And then some of the other items on this slide we have yet to cover. The next item on our list actually is to start to talk about antidiuretic hormone. So here we can take a different view of the effects of angiotensin 2 in the body, the RAA pathway. So again, we start with the liver. The liver produces angiotensinogen, which, look here, angiotensinogen is a small protein, that 453AA, that tells us that it consists of 453 amino acids. That's a pretty small protein. And when angiotensinogen encounters renin, renin is going to catalyze the conversion of angiotensinogen into angiotensin 1. And look at that, angiotensin 1 is a peptide, a small peptide. It has 10 amino acids. It too is inactive. When that angiotensin 1 passes through a blood vessel, particularly pulmonary blood vessels, angiotensin 1 encounters angiotensin converting enzyme, and it is transformed from angiotensin 1 into angiotensin 2. And oh, look at that, angiotensin 2 is an even smaller molecule consisting of 8 amino acids. Angiotensin 2, as I've mentioned before, we've talked about it previously, has many target tissues. And here, one of the target tissues of angiotensin 2 is the kidney. Another one is the adrenal cortex. And both angiotensin 2 and aldosterone promote the enhanced reabsorption of sodium and chloride, which leads to enhanced water reabsorption and decreases urine output. And that has the effect of holding onto body water to maintain blood volume. Another target tissue of angiotensin 2 is the smooth muscle in the walls of blood vessels all around the body, causing vasoconstriction. Vasoconstriction leads to higher blood pressure. So in this way, the RAA pathway works to hold on to body water and raise blood pressure. Previously, we've talked just a little bit about cardiovascular disease, and one of the most prevalent forms of cardiovascular disease is hypertension, or having chronically elevated high blood pressure. And the goal in treating hypertension is obviously to lower blood pressure, to keep blood pressure within the normal range as often as possible. And there are many different classes of drugs that are commonly prescribed to treat hypertension. And one of those classes is known as ACE inhibitors. So there are several different drugs that are in this class known as ACE inhibitors, but what they all have in common is they work to inhibit the activity of ACE. They slow down the action of angiotensin converting enzyme. So here we're looking at the RAA pathway, only we can easily see what happens when ACE is prevented from doing its job. So we can just fast forward, we're in the middle of the pathway here where we have angiotensin 1, and angiotensin 1 is an inactive molecule. In order to become angiotensin 2, it needs to be acted upon by ACE. But an ACE inhibitor, here's our red X, an ACE inhibitor prevents angiotensin converting enzyme, prevents ACE from converting angiotensin 1 into angiotensin 2. And if we can prevent the formation of angiotensin 2, we can prevent its blood pressure raising tendencies. This lecture is being recorded in April of 2020, and we're in the middle of the COVID-19 pandemic. And some preliminary research indicates that angiotensin converting enzyme, a particular version of it called ACE2, may play a role in the infectious ability of the virus. The way viruses work, all viruses need a host cell in order to reproduce. A virus needs to get its genetic material into a living host cell, and then that genetic material takes over the reproductive machinery of that host cell and gets it to produce new viruses. And so some preliminary research indicates that coronaviruses, one way that they're able to get into our cells, into the host cell, is by binding to a membrane protein called ACE2, angiotensin converting enzyme 2. And if you recall, one of the most abundant expressions of angiotensin converting enzyme is in endothelium of pulmonary vessels. And so we wonder if one of the reasons why coronaviruses lead to, at least in some people, this particularly severe respiratory response is because cells in the lung are most vulnerable to infection because they express so much angiotensin converting enzyme. And we're also trying to figure out why people with hypertension tend to be more vulnerable to the severe effects of coronavirus. And one hypothesis right now is that some of these people with hypertension may be taking an ACE inhibitor. They may be prescribed an ACE inhibitor, a drug, in order to treat their hypertension. But in response to this ACE inhibitor, cells can upregulate ACE. And so under the influence of an ACE inhibitor, seems a bit ironic, but the cells might express more ACE. And if you can think of this ACE2 as a potential entry point into our cell, taking an ACE inhibitor may, this is just a hypothesis right now, but it may make our lung cells in particular extra vulnerable because there is an upregulation, a higher degree of expression. More of this protein is present in the plasma membrane of people who are taking an ACE inhibitor than people who are not. The third hormonal mechanism we'll talk about is vasopressin. Vasopressin is also known as anti-diuretic hormone. And ADH is released by cells in the posterior pituitary gland. ADH is another mechanism by which the body, the kidneys in particular, will try to hold on to body water. ADH is secreted in response to mild to moderate dehydration. And dehydration means that there's not enough water in the body. There becomes an imbalance in the ratio of water to solutes. There's not enough water and too many solutes. And so this leads to a change in our blood osmolarity. When we are dehydrated, our blood osmolarity goes up. Blood osmolarity is monitored by chemically sensitive cells that are located in the hypothalamus. And when they detect a higher than normal blood osmolarity, that leads to the secretion of vasopressin, or antidiuretic hormone, from the posterior pituitary. So we'll talk about 3 actions of vasopressin, and similar to angiotensin II, the actions of vasopressin all work together to hold on to body water and to raise blood pressure. So the first action of ADH that we'll talk about is facultative water reabsorption. ADH turns it on. So far, we've only been really talking about obligatory water reabsorption and ways to dial that up or dial that down. ADH will turn on facultative water reabsorption. ADH can also inhibit the activity of your sweat glands. So under the influence of ADH, you'll sweat less, and that's reflective of a trade-off. It might be okay to let your body temperature go up a little bit in order to hold on to body water. And then the third action of antidiuretic hormone is to cause vasoconstriction of systemic arterioles. And we've talked about that several times at this point as a way of raising blood pressure. Let's walk through the homeostatic response on this diagram. So looking at the top right, number 1, high blood osmolarity triggers the hypothalamic osmoreceptors. These are chemically sensitive cells. They respond to an increase in our blood osmolarity by, number 2, activating the neurosecretory cells in the posterior pituitary. And those neurosecretory cells secrete antidiuretic hormone, also known as vasopressin, which is technically a neurohormone. It's a signaling molecule that's secreted into the blood by these modified neurons. And once in the blood, antidiuretic hormone has several target tissues. Shown here at number 4, we see antidiuretic hormone is going to cause the kidneys to retain more water, which will decrease urine output. Antidiuretic hormone also inhibits sweat glands, so you lose less water across the surface of your skin. And antidiuretic hormone can also lead to vasoconstriction of systemic arterioles to help raise blood pressure. Our focus is going to be in the kidneys. What are the effects of antidiuretic hormone on the kidney? In response to ADH, the kidney engages in what's known as facultative water reabsorption. And this process occurs in particular cells of the nephron, cells that make up the late distal convoluted tubule and where that distal convoluted tubule leads into the collecting duct and then the entire length of the collecting duct. I'll show you an image in just a moment. And antidiuretic hormone is responsible for your kidney's ability to produce what's known as a concentrated urine. So we've all experienced that concentrated urine, that dark urine that we produce when we're dehydrated. It's concentrated because your body is holding on to as much water as it can. And so that urine is going to have a high osmolarity. It's going to have a high concentration of solutes relative to water. And in the absence of antidiuretic hormone, your kidneys produce what's known as a dilute urine. So that really light yellow to clear, lots of water, very few solutes. That's a dilute urine. We can very clearly define what's a dilute urine versus a concentrated urine. And our reference points, our reference value, is the osmolarity of blood. Recall that blood osmolarity is about 300 milliosmoles. And a dilute urine is defined as a urine that has a lower osmolarity than blood. So anywhere between 65 to 299 milliosmoles. And so when we have a lower osmolarity, that means that there's more water and fewer solutes relative to blood. So that's a dilute urine. And it'll tend to be lighter in color. So it'll be that lighter yellow, almost clear urine. That's a dilute urine. And then a concentrated urine is defined as urine with an osmolarity that's greater than the osmolarity of blood. So anywhere from 301 to 1200 milliosmoles is a concentrated urine. And a concentrated urine tends to be darker in color. It has relatively more solutes and less water than blood. Notice here, I want to point out that the osmolarity of your urine can be the concentration of your urine can be up to 1200 milliosmoles. So under conditions of severe dehydration, your urine can be 4 times as concentrated as your blood. And while it's not a genuine reflection of reality, you can think of, just as a coarse way to conceptualize it, you can think of osmolarity as how salty something is, even though we know that there are way more solutes in both blood and urine than just salt, than just sodium chloride. But if you think about a urine that has an osmolarity of 1200 milliosmoles, it has 4 times the solute concentration as your blood. So there are 2 questions here. Under what conditions is it homeostatically favorable to produce a dilute urine? And under what conditions is it homeostatically favorable to produce a concentrated urine? And I hope you're thinking about water balance right now. So when the body has excess water in it, it needs to shed that water. And it needs to produce a dilute urine that has a lot of water and relatively few solutes. And that's the circumstance that we talked about earlier, where A and P contributes to the formation of a dilute urine. And under what conditions is it homeostatically favorable to produce a concentrated urine? It's when our body is dehydrated and our bodies need to hold on to as much water as it can while still continuing to eliminate wastes. Those wastes have to be excreted from the body. They need to be discharged, and we can only discharge wastes with some water. So if we can discharge those wastes with just a little bit of water, we'll be making a concentrated urine. So earlier in this lecture series, I guess in part 1, really, in part 1, we talked mostly about obligatory water reabsorption. And in part 2, we've talked about how obligatory water reabsorption is flexible. So under the influence of certain hormones like ANP, like angiotensin 2, like aldosterone, the cells that occur in the wall of the nephron can either increase their reabsorption of solutes or decrease their reabsorption of solutes. And water will always follow solutes. That's why it's called obligatory water reabsorption. We'll always move water this way, and we can dial it up or we can dial it down, but it will always occur. Here we're going to focus on facultative water reabsorption. So in biology, when something's described as facultative, it means it can be adjusted to accommodate environmental conditions. So maybe you've taken microbiology and you've learned about facultative anaerobic bacteria, where they can change their metabolic pathways based upon the presence or absence of oxygen from their environment. So here, facultative means we can turn this on or off depending on what the body needs. And what turns on this process, known as facultative water reabsorption, is vasopressin, also known as antidiuretic hormone. So When vasopressin is in the blood, it causes a particular region of the nephron, the late distal convoluted tubule, as well as the entire collecting duct. It causes those portions of the nephron to become very permeable to water. It makes it very easy for water to leave the filtrate and return to blood. And the way those portions of the nephron become more permeable to water is by inserting water channels, also known as aquaporins, into their membranes. Aquaporins are transmembrane proteins that allow water molecules specifically to very easily cross the plasma membrane. And we've talked about water molecules crossing the plasma membrane before. It's always called osmosis, right? That's the mechanism by which water molecules pass through a semi-permeable membrane. And way back in the early days of the semester, when we were covering osmosis, we talked about two ways in which water molecules can cross a plasma membrane, and one of them is by passing through momentary gaps between these neighboring phospholipids. Remember, water molecules are everywhere. They're far more numerous than these phospholipids. And then a second mechanism by which water can cross the membrane through osmosis is through these water channels. And these water channels were described a few decades ago, and initially the idea was met with resistance by the scientific community. But 10 years later, the scientists who discovered and described aquaporins were awarded the Nobel Prize. Here we're looking at a portion of the nephron. These cells occur in the late distal convoluted tubule as well as the entire length of the collecting duct. And over here, this is filtrate, and this is 1, 2, 3 tubule cells, and this is a peritubular capillary. And when there is vasopressin present in the blood, when ADH is present in the blood, here's a molecule of ADH, it binds to a receptor on the basolateral cell surface, and it's a signaling molecule that binds to a receptor. So we can see a signal transduction pathway is occurring inside the cell. And the signal transduction pathway is what leads to a change in the cell's behavior in response to that signaling molecule. Well, how these cells respond to ADH is by inserting into the apical cell surface here. This is the apical cell surface. They have these storage vesicles here just sitting there, and these storage vesicles have molecules of aquaporins in them. And when these storage vesicles fuse with the plasma membrane, it effectively inserts aquaporins into the apical cell surface. So now this apical cell surface, this surface and this surface and this surface, becomes extremely permeable to water. And water will always move by osmosis, so water will always flow in the direction of the higher concentration of solutes. If we follow the arrows here, we see water leaving the filtrate. This water is entering the tubule cell. This water is leaving the tubule cell across the basolateral membrane and moving into the blood. So this water, when we were back here in the filtrate, this water was about to be part of urine, but now it's been reclaimed by the body. Now that water is in the blood and it will not be lost from the body as urine. This is a different artistic representation of essentially the same process. What I like about this diagram is that it has sort of a better visual representation of these storage vesicles that are just hanging out inside the cell. When vasopressin is not present, these storage molecules, these storage vesicles, I should say, just hang out inside the cytosol, waiting to be inserted into the plasma membrane. And these vesicles this happens to be aquaporin 2. So those tan rectangles or molecules of aquaporin 2 that are just hanging out waiting to be inserted into the membrane. And then under the influence of vasopressin, this vesicle is going to fuse with the apical cell surface here, and those molecules of aquaporin 2 will then become part of the apical plasma membrane and make that part of the cell very permeable to water. Notice that there's aquaporin 2 over here. There are other versions of aquaporins. Not that it's super important for us, but I just like to point it out. Here's aquaporin 4, here's aquaporin 3. And so this cell becomes very permeable to water and makes water reabsorption extra easy. So in addition to the presence of aquaporins, water reabsorption requires a concentration gradient, right? Water will flow where the solutes go. So if we want to get water out of the filtrate and into the blood, we need to have a concentration gradient of solutes that favors that movement of water. Water chases solutes. So this brings us to the second component, or the second necessary condition for facultative water reabsorption. The first necessary condition was vasopressin. We need antidiuretic hormone to make the cells very permeable to water. The second required condition is known as the osmotic gradient of the renal medulla. These are the two things that I just said. The formation of a concentrated urine has these two requirements. Number one, we need vasopressin in order to insert aquaporins into the apical membrane of the cells of the late distal convoluted tubule and the collecting duct. And number two, we need to have this osmotic gradient in the kidney. So this is a diagram showing the osmotic gradient of the renal medulla. So let me remind you about the geographical situation of a nephron in the kidney, so to speak. So I hope you remember that part of the nephron occurs in the renal cortex. And so in the renal cortex, we can see the glomeruli. We see Bowman's capsule, also known as the glomerular capsule. We see the proximal convoluted tubule. We also see the distal convoluted tubule and part of the collecting duct. And then in the medulla, we see different parts of the nephron. In the medulla, we see the descending limb of the loop of the nephron, we see the ascending limb of the loop of the nephron, and we see a lot of the collecting duct. And so when we look at the extracellular fluid in the renal medulla, we see that there is a concentration gradient in terms of its osmolarity. So if we take a sample of that fluid, let's start it here in the cortex. Let's take a sample of this fluid, this extracellular fluid, this interstitial fluid, and test its osmolarity. Well, you know what? It's got an osmolarity of roughly 300, just like pretty much every other extracellular fluid in your body. But when we start to sample fluid in the medulla, we see the deeper we get in the medulla, the higher the osmolarity of this extracellular fluid. And so here's this gradient. We go from 300 milliosmoles to 400 to 600 to 800 to 1,000. And so we hit in the deepest regions of the renal medulla, not adrenal, renal medulla. That extracellular fluid has a very high concentration of solutes. And so it has a high osmolarity. And so now imagine there's filtrate passing through this collecting duct, and the collecting duct is very permeable to water. Where the solutes go, water flows, and this ever increasing concentration of solutes is going to pull more and more water out of the filtrate as that filtrate travels from the cortex to the deepest regions of the medulla. As long as this collecting duct is freely permeable to water, water is going to leave by osmosis. And that's how we end up with a concentrated urine. Here, this droplet of urine is like a maximally concentrated urine. It has a concentration of 1,200 milliosmoles. So, under the influence of ADH, water is going to leave the collecting duct because of the osmotic gradient in the renal medulla. And this is how we get a concentrated urine. This is a different artistic representation of some of the same concepts. Here we see the renal medulla, and as we descend, as we go deeper and deeper into the renal medulla, we see the concentration of the extracellular fluid increasing. We also see as filtrate travels the length of the collecting duct, we see more and more water leaving the tubule and returning to the blood. Also indicated in this diagram is a little bit about the mechanism by which this osmotic gradient is established and maintained. And that is a really cool topic, and we are not covering it. If you're interested in it, I encourage you to look up how the osmotic gradient and the renal medulla is produced and maintained, but we are not covering it formally. You'll just have to take my word for it that it exists. By point of contrast, let's take a look at the late distal convoluted tubule and the collecting duct when vasopressin is not present. So this is going to lead to the formation of a dilute urine that has more water and relatively fewer solutes. So when ADH is not around, the cells of this portion of the nephron are not extra permeable to water. They're just regular cells. And so as we watch filtrate pass through this collecting duct here, extra water is not removed from the filtrate. So facultative water reabsorption is turned off. And we end up with a urine that has a dilute osmolarity. It has an osmolarity of about 65. Not shown here, but maybe you're wondering, hey Laura, why do we go from 90 milliosmoles to 80 milliosmoles to 70 and on down? Not shown here, but additional solutes can be removed from the filtrate here. And when we remove solutes, that's going to cause the osmolarity to go down. So in the absence of vasopressin, facultative water reabsorption is turned off, and our kidneys will produce a large volume of dilute urine. To reinforce what we've just been studying about vasopressin, let's just go through this homeostatic mechanism. So we'll start at the top with the yellow star. Some stimulus disrupts homeostasis by increasing the osmolarity of our blood plasma, and this is typically due to dehydration. There's not enough water in the body. The sensory receptors are located in the brain. They're chemically sensitive cells called osmoreceptors. They're located in the hypothalamus, and the hypothalamus is also the control center here. So in response to those osmoreceptors, the neurosecretory cells in the posterior pituitary will start to release antidiuretic hormone into the blood. And we've been most focused on talking about the effects of antidiuretic hormone on the kidney. ADH turns on facultative water reabsorption and leads to the formation of a concentrated urine, a urine with a higher osmolarity, so more solutes, less water. This helps the body hold on to body water, which will help restore homeostasis. This diagram essentially illustrates the same things. Take a moment and go through it if you think it's helpful for you. What is a diuretic? Some of you know the answer to the question. You don't necessarily have to take a physiology class to know what a diuretic is. A diuretic is a drug that acts on the kidney, and it causes your kidneys to produce a larger volume of dilute urine. So diuretics help the body shed water. And diuretics are a different class of drugs that are commonly prescribed to treat hypertension. And the theory is here that if we can reduce blood volume by shedding body water, we can help lower blood pressure. So there are many different types of diuretics, and they all act on a different region of the nephron. What they all have in common is they reduce, one way or another, they reduce the reabsorption of water. So they cause more water to stay in the filtrate and consequently be eliminated from the body as urine. Don't get intimidated by this diagram, please. I'm only using it as an example. I found it on a pharmacology website, and some of you have taken pharmacology. Many of you will take pharmacology. And so this is a diagram illustrating the different classes of diuretics and where on the nephron they exert their effects. And we're not going to talk about any of these. These are all prescription. Most of these are prescription diuretics. We will instead talk about two very common over-the-counter diuretics. We'll talk about how caffeine and how alcohol each act as a diuretic. They have very different mechanisms. It is a myth that a low dose of caffeine acts as a diuretic. So your friend might say, oh, I just had a cup of coffee. No wonder I have to pee. Caffeine is a diuretic. Well, that one cup of coffee probably doesn't contain enough caffeine to have a diuretic effect. But at higher doses, above about 300 milligrams of caffeine, it turns out that caffeine slows the reabsorption of sodium in the proximal convoluted tubule. And when we slow the reabsorption of a solute like sodium, we're going to necessarily slow obligatory water reabsorption. So here we're back at our very simplified nephron. And you remember during reabsorption, a solute is taken out of the filtrate and returned to the blood. Under the influence of a higher dose of caffeine, sodium reabsorption slows down, and therefore obligatory water reabsorption slows down. More water stays in the filtrate, more water leaves the body as urine. Alcohol is also a diuretic, but by a very different mechanism. It turns out that alcohol prevents the secretion of vasopressin from the posterior pituitary. And so without ADH, without vasopressin, that late distal convoluted tubule and collecting duct are not particularly permeable to water. And so less water will be reabsorbed and more water will be excreted in urine. So we've seen this diagram before, only here the actions of ADH have all been crossed out. Without ADH in the blood, it's never going to bind to its receptors. And when ADH doesn't bind to its receptor, it's never going to trigger this signal transduction pathway. And without this signal transduction pathway, it's never going to cause the aquaporins to be inserted into the apical membrane. And so this cell will not be especially permeable to water, and extra water won't leave the filtrate. So alcohol results in the formation of a large volume of dilute urine. So this is watery urine. Maybe you're familiar with some of these phrases. It's pretty common among the college crowd, breaking the seal. And while the symptoms of hangovers are not thoroughly understood, it's consensus that most of the symptoms are actually due to the resulting dehydration. So the prescribed protocol following any amount of alcohol actually is to drink water before going to bed and as soon as you wake up, to try to counter some of the dehydrating effects of alcohol. This is the most interesting man in the world. He looks like my father-in-law, so I love him. I don't usually drink the recommended 8 to 10 glasses of water a day, but when I do, it's after a night of heavy drinking. So, at this point, we've talked about the basic mechanism by which the kidneys produce urine. We've talked about several mechanisms by which the kidneys can adjust their activity to alter the balance of water and solutes. Now we get to talk about what happens during micturition. Micturition is urination. The urinary bladder is a storage vessel for urine prior to micturition. The wall of the urinary bladder includes a thick layer of smooth muscle, also known as the detrusor muscle. And in order for urine to leave the urinary bladder and enter the urethra, we require the relaxation of two different sphincters, the internal urethral sphincter and the external urethral sphincter. So a sphincter is a circular muscle that acts as a valve to control the movement of a fluid from one place to another. So when a sphincter is contracted, it's closed. The valve is closed and it prevents fluid from flowing through it. When a sphincter is open, those muscle cells are relaxed, and it allows that fluid to then pass through the valve. And so during micturition, the smooth muscle that makes up the wall of the urinary bladder, the detrusor muscle, needs to contract. And when it contracts, it's going to decrease the volume of the urinary bladder, and that's going to cause an increase in the hydrostatic pressure of urine against the bladder wall. So when we increase the pressure inside the bladder and we open up both the internal and the external urethral sphincter, that allows urine to flow from an area of high pressure to an area of low pressure into the external environment. Notice that the urethral sphincters are made out of two different types of muscle tissue. The internal urethral sphincter is made out of smooth muscle. And I hope you recall that smooth muscle is under involuntary control. So it's under the domain of the autonomic nervous system. And the external urethral sphincter, that's made out of skeletal muscle. And skeletal muscle is generally under voluntary control. The external urethral sphincter is formed by part of the urogenital diaphragm, the skeletal muscle that makes up the floor of your pelvis, of your pelvic cavity. And so during micturition, the smooth muscle of the bladder wall contracts, and both the internal and the external urethral sphincters need to relax. They need to open up. During potty training, for those of you who have kids, we were all kids once, during potty training, the child learns voluntary control over their external urethral sphincter. Here's one place in the body where the female body differs significantly from the male body. And we will come back to this when we cover the reproductive systems, but here we are in the urinary system. And we'll start with the panel that's on the left. The major difference between the male and the female body in terms of the urinary system is the length of the urethra. So in the male urinary system, and we're overlapping here with the male reproductive system, in the male body, the urethra has 3 different regions. When that urethra is passing through the prostate gland, it's called the prostatic urethra. When that urethra is passing from the prostate into the penis, there's this tiny little region called the membranous urethra. And then as that urethra passes through the penis, it's called either the spongy urethra or the penile urethra. So the male urethra has 3 different segments, and the female urethra has just one segment. It's relatively short. And we think this short length of the female urethra accounts for the higher incidence of urinary tract infections in women when compared to men. So bacteria get into the body by entering the external urethral orifice, and they can make their way up through the urethra into the urinary bladder and cause an infection. That seems to be more difficult in the male body because of the length of the urethra. Dialysis is a very common therapy for people who have lost, to some degree, have lost kidney function. You can't live without at least some kidney function. And dialysis is a therapy that can prolong the lives of people who are suffering from kidney disease and can keep people alive until they're able to receive perhaps a kidney transplant. So you're in a position to thoroughly understand how dialysis works. We're going to talk about 2 common dialysis techniques. We'll talk about hemodialysis as well as CAPD, and I'll give you those details in just a moment. It's important to recognize what they both have in common. So remember, the goal of dialysis, which is the goal of your kidneys, is to remove waste products from the blood. And so in order to remove waste products from the blood, we need a semi-permeable membrane, and we'll need to have a concentration gradient that will force those waste products to move in the direction that we want. And so that concentration gradient is generated by this solution known as dialysis solution or dialysate. So let's start with hemodialysis. Hemodialysis is an older version of dialysis treatment, and it involves the patient going to a dialysis center and spending several hours there, potentially several times a week, depending on the level of kidney function. So the more impaired the person's kidney function, the more time they need to spend doing dialysis. And so in the dialysis center, the patient gets hooked up to a dialysis machine, and the patient's blood is removed from the body, passed through the dialysis machine, and then the patient's blood is returned to the body. So let's take a look at what's happening in this dialysis machine here. This is obviously a schematic diagram. It's meant for teaching purposes. And so inside this dialysis machine, there are many different small tubes. And these tubes the walls are made out of, it's called dialysis tubing. It's a semi-permeable membrane. And these tubes are surrounded with dialysate, this fluid that has the chemical composition required to draw waste products out of the blood. The second type of dialysis is called continuous ambulatory peritoneal dialysis, or CAPD. Continuous means it's ongoing. Ambulatory means the patient can walk, the patient can move around. Peritoneal refers to the peritoneum, a serous membrane that lines your abdominal pelvic cavity. And in CAPD, a dialysate fluid is delivered through a catheter into the abdominal pelvic cavity. So it comes to, we can see here, that dialysate fluid comes to occupy the space between the exterior wall of our visceral organs, like maybe this is large intestine right here, and here's the inner wall of our abdominal pelvic cavity. And if you recall from anatomy, the visceral peritoneum coats the exterior of our organs in our abdominal cavity, and the parietal peritoneum lines the inner wall of the abdominal cavity. So where this yellow is here, this is the dialysate, this is the dialysis solution, and it's coming to occupy the peritoneal cavity. And while that dialysate is in there, in that space, it has the chemical composition that will draw waste products out of the blood across that peritoneal membrane and into the dialysate. So we can see these little black arrows here are indicating the movement of waste products out of the blood and into the dialysate. And so this patient will leave the dialysate in their abdominal pelvic cavity for several hours. And during that time, they are ambulatory, so they can cook dinner, they can go and pick up the kids, they can do the laundry, they can be at work or be working. And after several hours, the patient then drains the dialysate out of the abdominal pelvic cavity through a different catheter and then discards it as medical waste. Here's a different diagram showing the principles of CAPD. So in general, treatment options, how do we treat a particular patient, is heavily influenced by their age, their overall health, and other aspects of their lifestyle. So let's consider dialysis treatment for a particular patient, and our choice is between hemodialysis and continuous ambulatory peritoneal dialysis. Which one do we choose? It depends. So think about hemodialysis. Hemodialysis requires that the patient travel to and spend many hours in a dialysis center. So it requires them to have reliable transportation as well as time. And that's not true for continuous ambulatory peritoneal dialysis, but rather the person who's going to choose CAPD needs to be able to take care of themselves. They need to be in good general health. They need to have good manual dexterity in order to be able to deliver dialysate and to empty that dialysate out from their abdominal pelvic cavity. The kidney transplant statistics are fairly depressing. So there are currently more than 100,000 people awaiting a kidney transplant, and that's just in the United States alone. And the median wait time for an individual's kidney transplant is 3.6 million years. And here's a statistic from 2014. In 2014, roughly 17,000 kidney transplants took place. More than 11,000 of those kidneys came from deceased donors, so an organ donor, and more than 5,000 came from living donors. So these are people who voluntarily give up one of their kidneys and then live out the rest of their life relying on the other kidney. There's great news there. So we've been doing kidney donations from living people for a number of decades now, and there appear to be no long-term consequences to health or longevity. So you can give up a kidney, and all things being equal, you will live out your full healthy lifespan. A developing technology is the artificial kidney, and there are several research groups trying to develop this technology. And one of the most promising is a collaboration between Vanderbilt University and UCSF. And there are links to very interesting videos about this technology. And in order to access the links that are embedded in these video lectures, I'll encourage you to go to the PDF of the lecture slides from back in the day, our original PDF lecture slides. And the link will be easily accessible there. So you can click on that and watch these videos. Here's a diagram explaining the basic technology of an artificial kidney. We hope that artificial kidney will be smaller than a coffee can, a little bigger than a soda can, because it needs to fit inside the abdominal pelvic cavity. And it relies on those same principles, right? We need a semi-permeable membrane, and we need a fluid that will encourage the movement of waste products out of the blood. So, in part 2, we focused mostly on how your kidneys are dynamic, how the processes of filtration, reabsorption, we didn't really talk too much about secretion in part 2, but mostly how filtration and reabsorption can be modified in order to allow the kidneys to respond to challenges in water balance. What happens when we have too much water in the body? What happens when we have not enough water in the body? And we talked about 3 different hormonal mechanisms for addressing water balance: AMP, the RAA pathway, and antidiuretic hormone. I described a little bit what diuretics are, and we looked specifically at the mechanisms of action of 2 different diuretics. We talked about caffeine and alcohol. We talked a little bit about the urinary bladder and the muscle contraction and relaxation that must occur during micturition. And then I gave you an overview of dialysis. If you have any questions, please reach out. Thanks for watching.